Hello, everyone, and welcome to another expert talk from Everything ALS. You know, we're working to detect early detection of ALS, and you can help us by joining our speech study project. It's just 5 recordings lasting about 15 minutes over 5 months. We need PALs and those with probable ALS and those who are healthy as controls. You can join us in finding the cure. And we would like to say that you can make a difference. Please join us because together we are powerful. Thank you. And with that, Christian, it's over to you.
Hi, everybody, and welcome again to another one of our Expert Talks series. Expert Talks. This week, we're being joined by leadership from Clene Nanomedicine, and we will have a conversation tonight with Marjan Sepassi, who's going to be presenting an update from Clene on their expanded access program and some related program materials. And I believe we're also being joined tonight by the CEO of Clene Nanomedicine, Robert Etherington, one of our good friends and allies in ALS. So with that, I'm going to introduce Rob and Marjan. I'll give a little background on our speaker tonight, Marjan. Marjan Sepassi is Vice President of Medical Affairs at Clene Nanomedicine, an experienced pharmaceutical executive with over 15 years of clinical medical strategy and health economics outcomes research and team leadership experience. Marjan led the medical strategy and field teams at Civi Biopharma, Arena Biopharmaceuticals, which is now part of Pfizer, and Actelion, which is now at J&J, in rare disease areas of high unmet needs. Marjan's entry into biotech started as an academic researcher focused on hematology oncology, and she holds a Bachelor of Science in Molecular and Cellular Biology from the University of Arizona and a PhD from the same institution. And Robert, I'm going to hand it over to you to introduce yourself. It's a pleasant surprise to have you join, but we were so glad you could join us. And I'm going to turn it over to you two now.
So thank you, Everything ALS team. Thank you, Christian. Thank you, Indu. A pleasure to be with you all. Marjan's the true expert. So Christian, your bio of her was far more relevant than mine. But I've been, here at Clene for 11 years now. I founded Clene as a founding CEO 11 years ago. Before we recognized, actually, that we were on an ALS journey, we were focused on neurodegenerative disease at the beginning and considered multiple sclerosis and Parkinson, but have since turned our entire focus to ALS, for which we are most grateful, and it occupies all of our waking hours because we certainly recognize that in this disease, all hands need to be on deck, and we are grateful that we can bring as many hands as we possibly can. Before this, I was 13 years at a rare orphan disease company called Actelion. When I was there for 13 years, and when we started Actelion, we focused on pulmonary arterial hypertension, which at the time was pretty uniformly fatal, save for a heart and lung transplant, and today is pretty well managed pharmaceutically. So we pray for a day, we work for a day, we hope for a day where ALS can be similar. Before Actelion, I was 9 years at Pfizer. So let's explain what we're doing. We are building a case across our programs for neuroprotection and remyelination with some very interesting ideas, but with one single asset, CNM-Au8. So this is an oral medicine. We have extensive patent protection on this and as many trade secrets. And we do all of our manufacturing in Maryland. Next slide, Marjan. What this asset is an oral therapy. So we take this by, not we, but people who are in our clinical programs take this by mouth every morning. And when they do so, they're drinking a gold nanocrystal suspension. And it can be equally administered by mouth or by feeding tube. It makes no difference. If I was blindfolded and I was drinking this asset versus my glass of water, I would not be able to tell the difference. In fact, all the clinical data that Marjan's about to show you is the placebo is purple-colored water. The asset is purple because of the way that light refracts and is absorbed, frankly, in the wavelength of light on the nanocrystal itself, of which there are many trillions in each 60 ml bottle. So this literally is unit of use dosing. As the patient or the person who takes this asset goes home, they are walking home with a little case of 30 of these, and that constitutes 1 month supply with 60 mils taken by mouth each morning. And this asset goes into the gut, covers the rather is encoded by a serum albumin protein corona that enables bloodborne transport and goes across the blood-brain barrier, targeting energy metabolism and oxidative stress. And I'll talk about that a little bit more. But first, there was a question. Somebody asked if it has dye. It has no dye. The drug asset has no dye. The purple colored placebo is food dye colored so that it could look the same as this asset. This asset is the nanosuspension at its concentration reads as purple. But again, it has no dye. It's literally a clean surface. That is to say, no chemistry whatsoever in a water suspension of nanocrystals that at concentration as they are, my eye reads it as purple. It's the way that light is absorbed and then reflected and refracted off the gold nanocrystal. But again, there is no dye in the medicine. There's only 3 ingredients, a lot of water, gold nanocrystals, and a small little piece of sodium bicarbonate, baking soda, effectively, that we use as a processing agent early. That is it. I'm drinking a clean surface nanotherapeutic. And we've shown you here on this screen a cartoon to reflect what is going on. So let me talk about that for a second. So your brain and mine is only about 3 to 5% of our body weight, but it consumes about 25% of our daily energy requirements. And that is going on day and night to keep my central nervous system active, responsive, and working as we pray it continually does. The ways that I move and walk and talk and eat and chew and breathe, all of this is this grand choreography of neurons that are doing what they need to do unless they're assaulted by, in the case of ALS, such a devastating, remarkably devastating disease. And what our asset's doing is improving nicotinamide adenine dinucleotide levels while reducing also, too, a byproduct of this energy, reactive oxygen species. And why this is relevant is because we are enabling the mitochondria to work more effectively against these pathogenic insults that ALS may insult, and to enable the better function and survival of that neuron. And how that's occurring is these nanotherapeutic suspensions by mouth orally that are crossing into the CNS are acting as an electron donor and receiver. So if we think back to maybe high school chemistry, what does a catalyst do? It lowers an activation energy to achieve a different energy. I used the metaphor earlier about how highly energy dependent my brain is, because to keep that mitochondria happy, effective, and working, there's a lot that needs to go on in order to do that. And so what we are doing in the case of ALS is really working diligently to see if we can provide the energy response and the energy enabling aspect of this so that the neuron can take care of its own housekeeping or return to the housekeeping that we expect it to do. Next slide, Marjan.
Brian.
So somebody just asked about side effects. So we have now collectively across a lot of different programs in Parkinson's, MS, and in ALS, as you see on the right side here, a number of programs we've commenced, including expanded access programs for compassionate use. We have over 700 participant years of exposure now to CNM-Au8. We've had this drug in the clinic now since about 2016 when we started our phase 1 work. Maybe it was 17 now that I think about it. We have long-term treatment duration taken chronically since we commenced with the Healy and Rescue programs, and at the same time started compassionate use back in 2019 of 5.1 years. Our treatment emergent adverse events are primarily mild to moderate in severity and transient. If we showed you a table listing, then we have some GI, some headache. Occasionally people complain of nausea, occasionally diarrhea. but we have not a single related serious adverse event, which is what SAE stands for, across any of our clinical programs to date, not one. That is remarkable, actually, in pharmaceutical drug development, to have not a single serious adverse event that a physician has assigned to the drug in any program. We also have seen no temporal association of increasing treatment-emergent adverse events, that is the nausea, the headache, or the SAE incidents, the serious adverse events based upon exposure duration. What that means is the longer you're on drug, we actually see our side effect profile drop, not increase. And we have not a single NOAEL finding across any of our tox studies. Now that stands for no adverse effect level that we've seen. So by consequence, the safety argument of Clene, we're about to talk about survival in a minute, the safety argument of Clene has been pretty well established now across 3 diseases in multiple clinical programs. And let's go now to, we'll talk about trials and EAP later. No, we have not had any, I see. I was thinking of a side effect of muscle. That's a different question that the viewers asked. So we'll come to some of these questions later.
Yeah, Rob, I'll jump in and say, feel free. We can do Q&A at the end if you want to get through presentation first, and because we're collecting all of them in a document, we can read them back to you as well as we go.
Super. Okay, great. Thank you. Yeah, that works nicely. Thank you. So this slide shows basically across, and we're going to focus on ALS now the rest of the time. We have data in Parkinson's and MS, but we're not showing it in this presentation. We're going to focus, of course, for the reason we're here, which is to talk about ALS. So Rescue was a 9-month study that we commenced in Australia. The Healy program is a 6-month study with James Berry, Merit Sudovich, Sabrina Paganoni, and others at the Healy program based in Harvard, alongside more than 50 of the clinical programs USA-wide that we organized across the ALS Healy platform study. And then, of course, we're also going to talk about our compassionate use real-world experience as well. We missed the primary endpoint in Rescue, and we missed the primary endpoint in Healy. That is, unfortunately, the case in many pharmaceutical drug studies of late in ALS. Too many to count, devastating all of us. We missed a Munich's endpoint in Rescue and an ALS-FRS endpoint in Healy. Now, let's talk about the studies for a second, because Marjan's going to give you a more deep dive approach, but I'm just laying out the full scope of these. The Rescue study was early to mid-stage patients. The average diagnosis was only 4 months. So these were participants that were quite new in their disease, and that was a 45-patient study, again, as I said for a minute ago, 9 months long. Munich's, it stands for Motor Unit Index Change. So what we were doing is we were looking at change in 2 muscles in the hand, the biceps in the arm, and the tibialis anterior in the leg. We were trying to ascertain if in these early ALS patients, of which about 70% were limb onset from their spine, 30% were bulbar onset ALS, but again, early, if we could influence the ability of the muscles of the hand, arm, and legs to change. We missed that primary endpoint primarily because in the early nature of this design, 4-month diagnosed patients, the bulbar subjects, which constituted nearly a third of the patient population, they never had any migration to limb engagement. In other words, their hand, arm, and legs in the placebo patients were completely normal. And so we were only assessing electrophysiological change in the muscle for 30% of our patient population in a place that they had no compromise. So we missed the primary endpoint. But we were surprised, again, when we consider all comers, limb and bulbar, to see that we had a decrease in clinical worsening that continued robustly in the open label extension, including with the people who converted to active drug as soon as they could. And we saw a preserved function in OLE. And we also saw some trends in some biomarkers. Critically, you're more familiar with Healy program. We were regimen C. That was a 6-month program. We missed ALS-FRS as the primary endpoint, as has, unfortunately, every one of the Healy participants to date. But we saw in that study a survival benefit. We saw in the double-blind program, we saw a decreased time to clinical worsening in the double-blind program, and we saw a neurofilament reduction in the double-blind program. Incidentally, neurofilament wasn't collected in the rescue program. That was started kind of before we started recognizing the criticality of neurofilament as a possible biomarker. In the open label of Healy, we had the opportunity to continue participants for another year, and we were able to add more biomarker assessments there, all of which we've seen some interesting results in. And then finally, we have 3 compassionate use programs that we've started to date. We started our first compassionate use program at the time that we commenced Healy, and that was funded in joint effort between the Harvard Healy program and the Clene itself. We have actually 84 people that we've been able to fund so far in EAP-01. Our second compassionate use program we call EOP-02 that the FDA approved was for non-Healy participants at various clinical sites across the country. Clene has funded that entirely on our own, and we have been able to enroll so far 211 participants there. And then finally, we received the NIH grant money, and that enabled us to start enrolling people earlier this year in the summer, roughly. And that is going to be a total enrollment of 180 participants, of which we've enrolled so far about half of them. So let me turn now to slide 8 and have Marjan continue.
Thanks, Rob. I'll start by talking about a key biomarker of disease progression in ALS patients. As many of you know, NFL, neurofilament light, is a cytoskeletal protein that provides structure and support for the cell. It's highly specific for neurons. As the axon receives damage, we have high release of neurofilament into the system. High neurofilament means what? We've got a lot of damage. High NFL in ALS predicts disease severity and prognosis. So with that in mind, I'll talk about some of the work that we saw with NFL in our Healy program. So we examined NFL in the blood as a biomarker of disease and disease progression. Essentially, what are we looking to do? We're looking to connect the mechanism of action, which in our case is the mitochondrial and neuronal health, to clinical benefits. Okay, so looking at the data from a long-term perspective, including double-blind in the Healy program, the green line, which is the CNM-Au8 treatment group, you see an initial separation during the 24 weeks. It's obviously nominal and not significant, but as you continue to look at the participants over 76 weeks, you see a decrease of 7.2% over time. Now, looking at the placebo group, which is the gray dotted line, initially you see an increase in the NFL, which stabilizes during the double-blind period and then continues to decrease as you look further past 70+ weeks. You have a delta change, if you will, of approximately 17.5%. It's important to note here that post the double-blind period, all participants are taking CNM-Au8. And then some of the clinical endpoints that Rob mentioned, I'll go over with you all, is initially the delayed time to clinical worsening as seen in both programs. So in the rescue program, delayed time to clinical worsening was defined as the first occurrence of death, tracheostomy, feeding tube placement, or NIV initiation. Again, here you see concordant differences between CNM-Au8 treatment and placebo in favor of CNM-Au8. On the right, you see delayed time to clinical worsening as defined in the Healy program. Slightly different definition here. You have first occurrence of death, PAV, tracheostomy, or feeding tube placement. I'm going to remind folks from a Healy perspective, many of you are familiar with our top-line data in terms of survival. We saw a great survival benefit with CNM-Au8 therapy in the Healy platform. On the left, you have a 98% risk reduction of time to death or PAV, and on the right, time to death also has a 98% risk reduction in favor of CNM-Au8. As I move on to the long-term survival analysis, I'm not a biostatistician, but I'll talk about propensity matching. In absence of concurrent placebo groups that we can compare survival for in our programs, we use propensity matching. It's used a lot in other disease areas, and it's used in ALS for long-term extension programs such as this, and compassionate use programs, as I'll talk about later. And essentially, this is when we are missing the placebo-controlled arm. And what do? We match each person to the best of our ability based on key disease parameters at baseline that we think are significant in ALS. So in this case, as some of the examples are age at onset, site of onset, whether it's bulbar or limb, TRICALS risk score, etc. Now, there's many benefits to conducting the propensity matching, but there's also some limitations. We can account for the variables and covariates that we know of as of today, but there are unknown differences that can affect our analysis. So that's why we strictly rely on placebo-controlled trials for accurate and full data analyses. However, without the absence, we are able to use propensity matching. And it works better when you have larger samples. And the reason it works for us in ALS is that we have historical clinical trial data in the name of PROACT that we are able to utilize free of charge across the scientific community to use the placebo arms of folks that participated in clinical trials in the past. We also have the ALS natural history data set, which are a registry of folks that are being seen routinely in clinical practice. We also utilized MND Australia's database, which is specifically for the rescue comparison, as Rob alluded to, which was conducted out of Australia. So looking at the comparisons to look at the survival benefit for CNM-Au8, we look at the controls. Regardless of the control that we see, we have a good survival signal in favor of CNM-Au8, whether you are comparing to PROACT database or the natural history consortium controls. So folks that were prior in clinical trials or those that are routinely seen in clinic.
Marjan, there was one request. Could you maybe shrink a little bit the left-hand column so the slides could be a little bit bigger?
Sure.
Thank you. Hopefully that helps a little bit. Okay.
Does that help a little bit?
Thank
And
you.
also you can put the toolbar up.
I think it'll probably go off screen if she does that. So no, we're good. Don't worry about it. Thank you, Marjan.
Thank you.
Sure. Sorry, it was frozen and would not let me advance in the presentation mode.
Marjan, one more thing. There's a little arrow at the top of in the toolbox. If you push that, it might get that out of the way and make it a little bit bigger. It's on the far right side.
Okay. I moved it to the bottom. Did I press the wrong thing, Rob?
It's the little flying wedge in the bottom right corner of the ribbon up top.
Right here?
Yeah, let me try this, Marjan. Maybe stop sharing. Let me see if I can go to share mode. I might be able to.
Yeah. Maybe
we have it.
Oh, there we go. Yep.
Thank you. Much better.
All right.
So essentially, in both analyses, we saw a important survival benefit compared to natural history, whether we're looking at PROACT controls or the natural history consortium. Next slide. Now I'll shift gears to talk about our expanded access programs. Rob alluded to a lot of the information that I'm presenting here. We've started our expanded access programs concurrently within our development program, which isn't very common in the development world. The first expanded access program was a single site program out of Mass General. It enrolled 79 patients. And the second expanded access program was offered to all Regimen C Haley ALS platform trial sites to prioritize those folks who had participated in the OLE. As a result, you can see 22 participants rolled over to the expanded access program. So they participated in Healy double blind, they completed the open label extension, and then they moved over to expanded access program. Of the 36 sites, 18 sites opted to participate, which is a wonderful showing. As of now, we have approximately 300 participants who are on CNM-Au8 since 2009. We have some folks who have been, as Rob said, on therapy for over 5 years. And we're really committed to continuing these expanded access programs. And I'll show you why they're important and one of the main reasons that we're here to discuss with you today. Looking at the survival for the expanded access program, again, if we compare to natural history, because we don't have placebo arms, as we talked about earlier, again, you see a survival benefit in favor of CNM-Au8, whether you're looking at the natural history data set or PROACT. Next slide. One of the most important programs that we have ongoing at the moment at Clene, in partnership with Columbia University, Synapticure, and our team, is the NIH-funded EAP. We were one of the first programs to be funded for a course of 4 years to have an expanded access program in partnership with these institutions. Next slide. So, as Rob mentioned, our goal is to provide investigational product, here CNM-Au8. Our grant, it says 100 participants diagnosed with ALS. When we submitted the grant, our goal was 100 participants. As we initiated the program and we did our best, we've been able to reduce costs and streamline our operational infrastructure. That's why you heard Rob say approximately 180 participants. Because of reducing our costs and streamlining our operational infrastructure, we've been able to maximize our funding received by the NIH, and we hope to enroll almost double what they gave us the grant for. As you all know, compassionate use is saved for those who do not qualify for clinical trials. Essentially, it is for drugs that have good preclinical data and are in clinical trials, but there is a large portion of the population that does not qualify or meet the inclusion criteria for clinical trials. So as a result, we have this program. We look to evaluate the safety and efficacy. I'll talk a little bit more about that as we look at the data for CNM-Au8 treatment in ALS participants. If you could go back once more, Rob, sorry. Our funding allows for up to 144 weeks of treatment. As it currently stands, everyone will take CNM-Au8 orally or by feeding tube. Our aim is to have a balance of what you see in a traditional clinical trial and then the convenience of remote engagement. I think this is important to mention as we try to maximize where we gather data, including real-world evidence. So at some of the clinics, the 12-week visits will be required to be conducted on-site, and other 12-week visits can be performed remotely. As Rob mentioned, and maybe you didn't actually, as part of the program, one of our biggest aims was to have access to those who are interested in participating in programs, but they live very far from research centers or are in rural areas where they do not have access. So that is why the partnership with Synapticure is very key for this program. And they're able to participate 100% virtually. Next slide. So we will be collecting safety information as we always do for expanded access programs, but this is one of the first times in the ALS research arena that we're using real-world data in order to move the needle, go beyond past providing product on a compassionate use base, but also collect efficacy information and understand more about CNM-Au8 in a real-world setting and utilize that data in a larger population. So we, from an overall holistic perspective, understand CNM-Au8 better. So as a part of that, we'll be assessing breathing assessments. We'll be conducting ALS-focused questionnaires. We'll be collecting blood to continue to look at biomarkers and beyond, as well as conduct safety assessments. Next slide. So I'm proud to say we are on target for our expanded number beyond the 100 and that we were approved. We will have 180 participants by the end of the year. As of today, I even have a more updated number than Rob was able to give a little bit earlier. We have 103 participants that have enrolled and been dosed CNM-Au8. We've been successfully expanding the program into rural areas across the U.S., which we are grateful for our partnership with Synapticure. I can say there are participants in Alaska that are hundreds of miles away from a nearest center. In fact, we have to fly staff in order to be able to conduct some of the lab work that is needed to be part of the program. As I mentioned, the importance of long-term data collection, we're moving the needle. Can we start to utilize these expanded access programs and real-world data to better understand our products and hopefully both in the regulatory framework, but also in our future, hopefully, access of CNM-Au8 once it's in the population. In terms of centers, we have 7 brick and mortar sites across the United States. We have Duke, Nova University, Penn State, Northwestern, Texas Neurology, and University of Idaho. We also have, as I mentioned, our telehealth clinic, which is the Synapticure Teleneurology Clinic, where half of the patients allotted to the NIH EAP will be fully remote. I know the remote is of great interest, so I'll talk a little bit about where we stand with Synapticure. This is their data, so I thank them for sharing it with me. Next slide, please, Rob. So Synapticure is a multidisciplinary clinic that operates across all 50 states. It's an extension of care for ALS centers and supports patients who are unable to access EAPs and at traditional sites. In the 5 months since the program has been active, 72 participants have been enrolled and 15 are scheduled to screen by mid-December. So that means of the 90 or so, which is about half the population of the EAP that are enrolled or in screening for the study, 30% of them live in neighborhoods scoring in the top third of the most socioeconomically disadvantaged. We have an average of 180 miles from the closest NEALS affiliated clinic for those that are enrolled. Many of the participants are new in the world of ALS-related clinical trial research in general or involvement in any program. As of now, from a demographics perspective, 42% are female, 10% Hispanic or Latino, and about 13% non-Caucasian, which we all know from the clinical trial arena that is usually not the case. And then I'll hand it to Rob to talk about next steps. I know folks will have a lot of questions regarding the EAP programs and our next steps, so I'll let him finish off and then we'll take those questions. Thank you.
So moving from right to left in this case, so obviously Marjan's just spoken about our need to collect and report the long-term ALS experience. That occurs not only in the compassionate use EAP programs, but also in the open label extensions from Healy and from Rescue. We are continuing to follow the many participants that kindly gave us their time and participated with us in these 2 clinical programs. And we are grateful to have enabled them long-term access on our open label extension in those programs. And we are very grateful that they're continuing to come in and to be ascertained clinically so we can report to regulatory agencies worldwide that long-term experience critical for ALS, especially as we are studying, in Clene's case in particular, survival and survivability. Now, in that sense, too, we recognize that we will need a global phase 3. So independent of regulatory paths for pretty much any market worldwide, there is a need to commence a phase 3. We've already been public in earlier forums that we're planning to commence first patient, first visit into our phase 3 program next year. We are, in fact, organizing that protocol path and the regulatory approval of same with the bodies that matter in the EU, US, North America generally, and other parts of the globe presently. And we are also planning to raise capital to fund these phase 3s next year. Now, that process and that phase 3 is comprehensive and large and by consequence expensive. And so in that spirit, we also have already been public that we've announced earlier this year that we are presently actually talking to the agency to understand their view on the possibility of approval and regulatory pathways forward. So in the spirit of that, we announced earlier this year in the late fall that we had a meeting that we've organized for this quarter with the FDA's Director of the Office of New Drugs, as well as the Director of the Office of Neuroscience and the Neurology Review Team that is in charge of reviewing our asset. And that meeting was planned to be with key experts, 3, and ourselves at the executive team, and we'll be announcing the outcome of that meeting after we receive and fully digest the minutes from this meeting from the agency. And as Clene has done in the past, we will be explaining this roadmap that we envision from that regulatory interaction in a future press release that we envision will be this quarter sometime. So that's obviously this month. In other words, now we're at the end of the quarter, as Clene has previously announced, and I anticipate that that will still be the case. So let us turn now, with gratitude for your time, to other questions that have been raised. And Christian, do you want to, I'm not sure how best to approach the questions. Which is the right approach?
Sarita and I will, thank you for that, Sarita, and thank you both for the presentation. That was really helpful, and you covered a lot of ground, so very, very grateful for that. Sarita and I will kind of go back and forth here, curating the questions for you. So we did cover a couple up front, and I think we've covered reasonably well when the drug will be available, that it's available now in EAP. There were some related questions around the function of the drug first. So I'm going to jump in with, how effective is the transfer across the blood-brain barrier orally? How has that been observed so far?
Marjan, do you want to take that? You're on mute, I think, Marjan.
Yeah, you're on mute. There we go. Let's see if I can. Did you get a little prompt to ask to unmute? Sarita, could you help her out with that, or
Marjan says that you're still on mute from our side. So let me just jump in while we solve the Marjan mute thing. So we've seen evidence of our nanotherapeutic suspension actually through high-powered MRI machine in multiple tissues of the CNS, including the eye, in the brain, in the spinal cord, and also in neurons even. We've seen examples of our nanocrystal. Exactly the percentage of BBB translation, we don't have that percentages structured, but we know that we see BBB cross so that we can actually have effect. And we did actually 2 important studies in other neurodegenerative diseases we call the repair programs using a 7 Tesla MRI machine. So that's a very high-powered MRI. If I sprained my ankle, I would be in front of an MRI machine that would be 1.5 Tesla. There's only a few 7 Tesla systems that are that powerful across the country. And we did this to actually see if we could ascertain energetic metabolite change within the brain of MS and Parkinson's subjects. We tried to do ALS, but the amount of scanning that is necessary in this particular clinical program requires that the patient be supine, that is laying down on one's back for an hour. And there were many ALS patients and ethical boards as well that thought that that was not possible without any breathing apparatus support at all. So we don't have the data for ALS to show this type of a BBB cross, but we did see it in MS and Parkinson's.
Thank you. The next question is, thank you, Marjan and Rob, for doing this. The next question is, have any participants reported muscle response or recovery?
You're talking about, I think, some functional improvement by muscle. That's what I'm presuming that means. So what we saw actually in the limb-only onset of the rescue program, and again, I mentioned that there was a pre-specified endpoint of both bulbars and limb onset, and yet we were assessing hand, biceps, and leg function in both subjects. We didn't see any bulbar change in muscle because the placebo bulbar subjects had no compromise in muscles. Their hands, arms, and legs in their early nature of the ALS disease was not compromised. They received, bulbar compromise in the way they spoke or the way they swallowed or chewed or were able to, manage all the musculature in the bulbar region, but not in the limb onset. However, this is a very important point. In the limb onset patients, even though we were well underpowered, we did see Munich's change there. In fact, the way our modeling suggests, if the study was a limb only, that is, we had excluded bulbar patients, which we didn't do in either of our programs, we may have even met the primary endpoint there. The other, so that in that respect, it was an interesting muscle improvement. The ALS-FRS result that we saw in that study was trending quite favorably, and there is a muscle component of ALS or ALS-FRS, as well as in the open label extension of the Healy program. But the primary benefit we've seen, as we've stated already, is with respect to the hard endpoint of survival change.
All right, thank you. And next one is, are there any plans to expand to the UK and Europe? I guess, or
yeah, so the phase 3 is going to be a global phase 3. So that's the primary way that will happen. So we intend a global phase 3 because we, frankly, of course, need this data to commercialize worldwide, which is our intention. The primary way to do that is, of course, only through the phase 3. There's not, unfortunately, compassionate use programs in most of the countries in Europe that we could enable. In any event, Clene does not have the ability to fund those extra initiatives. In fact, we're, frankly, the reason we leaned into the NIH grant and very much thankful to a bipartisan Congress who paid for this, is that enabled us to expand when we didn't otherwise have the ability to expand further ourselves. These are extraordinarily expensive programs. And Clene presently for EAP 01 and 2, well, 02 is paying for everything and 01 with Harvard. And so it would not be possible for us to continue paying for expansion in EU or UK. But the phase 3, we will, to the point.
Thank you. You met with the FDA on November 1st about accelerated approval. What was the result?
We haven't announced the dates of this meeting actually, but we will be, as I mentioned a minute ago before I started this Q&A, we will be releasing the data, or rather the path forward after the agency gives us the full minutes from these meetings, and we are able to work with the agency on the path forward. So we do expect such announcement will be this quarter.
Thank you. You mentioned that the results when missing the primary endpoint were a result of kind of the mixture of participants in the last phase of study. What have been the observed, drilling down a little bit, what have been the observed motor effects of CNM-Au8 on PALS participants with bulbar symptoms? Could you call out that specific subpopulation's results?
No, I can't because, again, as I mentioned in rescue, during the 9 months, the bulbar specific individuals did not have any muscle compromise during that early stage of their ALS disease. And that was the only call out we did specific to bulbar in a functional capacity. So we don't have other data from Healy, for example, that shows in bulbar only subjects improvement muscularly, because in that data set, we have from Healy the entire combined data set of both ALS, bulbar and limb onset.
Are you able to share any, in that same vein, are you able to share any results for any other subpopulations you identified by way of the ALS-FRS against any specific modality or vector of the ALS-FRS, where you saw greater responsiveness, like maybe by endpoint, either speech or upper limb or lower limb response?
No, we actually are right now analyzing all that data set. Harvard has just given to us, the Harvard Healy team has just given to us all of the data so that extra work of subgroup analyses can commence. But thus far, to the point, Christian, we've been primarily looking at the primary pre-specified secondary and exploratory endpoints, not subgroup analysis extensively yet.
Okay. Thank you.
Thank you. Do you expect the FDA to approve your drug candidate? When and how? What's the plan to get this to market?
Well, that is exactly the question that Clene wants to know. It's a very important question. Excuse me, as we've already stated publicly, we've submitted all this data to the agency, and we have talked at length about the survival and the neurofilament biomarker achievement. So of the 5 now completed Healy programs, we are the only one that has showed in the double-blind phase survival benefit, as we just showed, and we're also the only drug in the Healy program that showed in the double-blind phase neurofilament benefit. We've also announced further open label and EAP survival that Marjan's just given you some insight to. More is contained on our website. And we've also released further data on neurofilament responders, even super responder group, and how earlier this year we announced at key scientific meetings how those neurofilament responders had better effect both functionally and with survival. So all of that is now going to the agency. The key question really in front of Clene particularly, but more broadly, everybody who cares about an ALS patient is to what degree is there regulatory flexibility? We did see this definitively with the SOD1 ALS asset tofersen. We did see this also with Amylyx, who had their approval based upon one study. So Clene has now Rescue and Healy, and indeed, as we've stated in this call, we missed the primary endpoint, but we've nonetheless seen hard endpoints like survival benefits, both in the double-blind period and throughout the open label, and as I said a minute ago, in compassionate use. So that is the key question. I think the regulators are now considering the same, and there may be regulators in other markets besides the US that also are having these same questions, and Clene looks forward to the opportunity to present these views, and that's why, in fact, when we plan for the interaction this quarter with the agency, as we announced in September, we brought with us key opinion leaders that are names that you would know to make the case. And so we'll see in the coming weeks what the agency feels about this and what would be the path forward that they would enable us to be on. So that still is very much a work in progress, this question. And Clene, more than as much as you, very much looks forward to this answer.
In that vein, I'm going to jump ahead. We had a couple of questions. I think one of them, well, I'm going to get back to one because it's a little bit more about expanded study outside of ALS. So for the person who asked that, I will get back to it. In the charts on slide 11, where you talk about percent survival rate versus time to major event, how does the data account for subgroups? You mentioned that you're still doing that analysis. Is part of that analysis study to look at subgroups, meaning slow progressors versus fast progressors, for instance?
Yes, bulbar versus limb is also a very important piece. And frankly, the subgroup analysis on neurofilament also has a bulbar versus limb component as well. And so this is actually all work that's presently underway. The slide 11 data you saw was the regimen data. So what we were showing there is the Healy all-comer presentation in the pre-specified secondaries. And that subgroup specificity wasn't laid out as the pre-specified piece. And that's really what the regulatory bodies want to see is what the pre-specified part was. But we concur, Christian, with you and everybody on this call. The subgroup analysis is important, but we have not yet concluded nor announced that yet all the way.
I'm going to add, we have a lot of our consortium members who present, and all these companies that Rob has described, that they struggle with the endpoints. It's really a challenge of design, right? Like it's hard to know which subpopulations are to define ahead of time in a trial design in a disease where so little has been properly defined going into these studies about what our subpopulations are. So doing the post-analysis is really one of the few ways to get there until we can get much more precise data into the system beyond ALS-FRS, like much more precise outcomes measures like we do with our observational studies. That's the reason we do those, is because we're trying to support companies like Clene that need even cleaner endpoints, and everybody, all of us involved, need better subpopulation definition going into trial designs so that the FDA isn't kind of calling companies to task after the fact when they're not really accepting a lot of subpopulation definition going in. So.
And the other challenge, and I think that we'll see this as we get deeper into reg discussions globally across the community, not just with Clene, but if there's too much subgroup specialization, then what we have is the challenge that the label is very specific to a subgroup population, and the insurers therefore could lever that in their reimbursement or not. So, so at the beginning, you know, there is utility for a broad a label as possible, which means that the clinical data is as broad as possible.
I think the other thing to keep in mind, Christian, is as you know, you design these phase 2 programs, these are safety and dose finding studies, right? So we are trying to get a lot out of the phase 2 programs without incorporating so much of these subpopulations. As Rob mentioned earlier, for the rescue program, if we had known in advance, would we have enrolled bulbar patients? Probably not, because our data would have been positive without the bulbar subgroup population. But again, for a phase 2 dose finding studies, it's hard. Those are big trade-offs to make.
That's the science, right? It's the learning as we go. I'm going to keep going with the community questions here. One really important one: what are the EAP participant criteria? ALS, FRSR, time from onset, et cetera.
Yeah, so there isn't a criteria for ALS, FRSR. Expanded access programs, by and large, and by definition, are compassionate use. So very little inclusion criteria. We have it listed on clinicaltrials.gov, but being able to do an SVC is one of the criteria. But by and large, very few inclusion criteria. ALS, FRSR, as asked, is not one of them. So it's for those folks that don't qualify for clinical trials.
If Marjan or Rob, if you could drop the link on where people can learn more about the sites that might be close to them, or learning more about Synapticure to be able to at least start their process, that would be, we'll make sure we can share that.
I'm happy to share, Christian. The only thing I want to be clear, is at this point, all the sites, brick and mortar and Synapticure, have reached that full capacity.
Got it. Okay.
So by the end of the year, we anticipate all participants will be enrolled and dosed pretty closely with CNM-Au8. So happy to share them, but this is sort of the conundrum that we face. There's only so many participants, and we've maximized at this point what we can include in the expanded access program that's funded by the NIH.
Well, we'll have to keep we'll have to keep making sure that this is a funded program with Congress in the coming year.
Not exactly the point, actually, is that as more economics are enabling, the Clene has the ability to fund more of this, including if we get more funding from the NIH, which is something that we're actively pursuing.
Thank you. So I'm going to get back to a couple of the other questions about the ongoing study. Do any of the participants have the SOD1 mutation? If so, do they use Qalsody, and is that a confounding factor, or is that a disqualifying factor in Clene's asset?
From the expanded access program, is that what you're referring to, Christian?
Yeah.
I'm sorry, I didn't get that.
Basically, are there any participants within the study? It doesn't have to be EAP. It wasn't really defined in the question. Have the SOD1 mutation, and if so, was their use of Qalsody considered, or were they not part of the study?
So the expanded access program does not exclude any particular patient group. However, there is a treatment that is approved for SOD1 patients. So we did have participants on background therapy, including in the EAP programs even now, as long as it is okay if it's an investigational product, it is okay by the other manufacturer. Clene, as part of the expanded access program, does not prohibit use of other either unapproved or approved products. However, for SOD1, there is an approved product which would not be included.
Just as an aside, that's an important point. So our drug doesn't have any drug-drug interactions with any other drug that we know of. It's used in combination with, regardless of what disease we're talking about, with whatever standard of care therapy the participants come to Clene on. So in the case of multiple sclerosis, we actually require that they be fully controlled on their MS drugs, their disease-modifying therapies, that were tamping down their immune system. The doctor could put them on whatever drug they wanted in MS, and they came to Clene. Similarly, that was the case in the Healy program in particular, and Rescue 2. Edaravone and riluzole were both background therapy. And then once the Amylyx was approved for a period of time that it was on the market, it was also used as well. And so Clene lets participants on all of these assets, and then Clene is placed on top of those drugs.
Okay. Does compassionate, I'm going to stay on the EAP real quick. There's a question about compassionate use. Does access via compassionate use, is that prevented by full enrollment by EAP, or are they separate and distinct, or are they the same program?
So compassionate use is essentially the same, is an alternate term for saying expanded access program.
Mm-hmm.
In the development program of CNM-Au8, we've had 2 that were predate the NIH funding. This is the first time that we've received NIH funding for the expanded access programs. So they are one and the same. It does not, compassionate use is essentially the same thing as saying expanded access program, and all of them are compassionate use programs.
Thank you. Getting back also to the mechanism. What is the specific mechanism in layman's terms does Au8 do to nerves?
I can take that. So in terms of, what we do from a product perspective, our theory, CNM-Au8 is not a small molecule. It's not inhibiting proteins. It's not a monoclonal antibody. It's not gene therapy. Essentially, what we're doing is simply permeating cells and providing an environment where NADH is shifted to NAD naturally, and we improve the mitochondrial function and ultimately improve neuronal health so that the neuron can survive and function better and longer.
Okay. And tying that to the neurofilament numbers that we've seen, do the neurofilament numbers decisively show decrease when on medication?
Yeah, so we've announced that actually not only double-blind phase, but also in a super responder group and subsequently an open label. And a key question that has been raised by the value of EAP is we're also, as part of the National Institute of Health protocol, which we have funding from the government for, the clinical advisors of this program and Synapticure recommended we test neurofilament there also from baseline and throughout. So we're trying to build a neurofilament robust data set. And this is also relevant because it's a possible avenue of a regulatory path worldwide. If the drug CNM-Au8 is able to drop neurofilament and that precedes a benefit in survival or precedes a benefit in function, does it tell us that patients who have a neurofilament response have a benefit of being on the drug? That's the key question that we're presently right now working to explore deeply.
And that question is probably driven as much by amount of time needed. One thing learned through tofersen and others where neurofilament's been used is that there's a delay, right? There's an observed delay of when neurofilament starts to go down after being on drug.
Yes, that's correct.
Mm-hmm. Let's see. We did have a lot of questions, so I want to make sure. I know a lot of them were kind of similar to each other, so I'm filtering through a little bit. Actually, one that I wanted to go back to was a good question about investigational studies. Have you considered investigational pilot studies in other areas of neuronal injury, like traumatic brain injury, anoxic brain injury, AP, cardiac arrest, ischemic stroke, et cetera?
So if I was to take my laptop 5 doors down to our head of translational medicine's office who plans this, we get queries on a weekly basis from clinicians or researchers asking us to consider such studies. That's the interesting thing about CNM-Au8. At the exact same dosage, 30 milligrams, we've now studied ALS, Parkinson's, and MS, including we have presently received protocols on 2 of the list you just gave me, and a few you have not in the neurodegenerative space. The key thing, though, for us to commence this is we need to understand the regulatory path forward, as well as work with our investors to get more capital to pursue these programs. But we do think we have a pipeline and a product, and we have pretty demonstrably shown the safety. And so that actually is why, from both an academic perspective, and if it's my disease, it's not academic at all, but I mean, from a clinical investigation perspective, we think that the risk of AU8, CNM-Au8, as we call our drug, is very low. And so the question is, how do we find enough resources to explore fully our ALS plus these you've mentioned? But there is an enthusiastic orientation of ours to look at a number of neurodegenerative diseases as possible.
Great, thank you. And I'm going to tie another question to that, which is putting our kind of squawk on the street hats on for a second. What is, how does that tie into a status of partnership discussions with larger pharmaceutical companies to help accelerate and fund phase 3? You're seeing a pipeline specifically in the ALS realm, but also in related areas, and there's a lot of movement in the space. Companies,
and especially there was a key announcement this week, another one last week, $1.5 billion of announcements in 5 days, actually, of neurodegenerative programs. So we actually have right now pharmaceutical companies looking at Clene. We welcome partnerships. I think it's the best way to explore to working with big pharma. Clene is a small little pipsqueak company with respect to our much larger big pharma possible partners. And so we have 5 that are right now looking at Clene in different ways under our hood, evaluating our data, and we welcome all such possible partnering conversations.
Great. Great, thank you. I believe we've asked, and I think we've asked it a few different ways in terms of any analysis, but I'll ask just to make sure we covered it. Is there any analysis, or is your post-hoc analysis including, as you look at the different subpopulations and effects, an analysis on upper motor neuron versus lower motor neuron effects?
Yes.
Marjan, you want to take that one?
Yeah, and in terms of, even from the rescue perspective, we do and will continue to analyze. But in terms of future, how it will affect the phase 3 program, not necessarily part of our initial analyses for the design of the RESTORE program, upper versus lower.
And Christian, I saw a question more than once here that we've not yet addressed. I want to make sure we get to it. Our primary mechanism is to increase NAD+ levels. That's what we've really seen. That's been the primary study. The translational work I mentioned earlier with Repair's 7 Tesla system was exactly expressly designed to show a statistically significant improvement in NAD+, which it did. And we have really leaned into this also, too, in our presentations to the agency and the briefing books we've sent them about how NAD+ and giving the neuron and the mitochondria in that neuron the energy it requires, one example of which is the energetic metabolite NAD+, is a key thesis of Clene's mechanism.
Thank you. That was exactly where I was going next. So thanks for jumping in, making sure we covered it. With that, we are at the kind of past the 8 o'clock hour at this point, East Coast time. So thank you so much for Team Clene, for Rob and Marjan's presence, and for addressing all these different questions. If there are other questions that come in, if the community has, Rob and Marjan are incredibly accessible, so we can pass them along as well. And we will, make sure that they see them and provide any answers back. Thank you for your dedication.
And thank you for everybody's dinner hour learning about this. We very much appreciate your time.
Yeah, it's great to have companies like yours. Like you said, you're not a big company, to be focused on ALS and doing everything you're doing and building partnerships and looking for expansion of a program that has a lot of potential for it and that we can continue to advocate for via NIH funding and other public sources. We're here to work with you as well. So thank you.
Thank you, everybody.
Thank you.
Thank you, everyone.
Thank you, Marjan and Rob.
Thank you. Have a good evening.
All right. Thank you so much.
Thank you.
Folks, we had to come to our living room again. It's like we save a lot of gas money, a lot of time, and we get a lot of good emotional support just by showing up. So thank you to each one of you